This Week in Virology, the podcast about viruses, the kind that make you sick. This Week in Virology, episode 191, recorded on July 13th, 2012. That's Friday the 13th. Hi, everybody. I'm Vincent Racaniello, and you are listening to the world's only podcast about viruses, TWiV. And joining me today from North Central Florida, a return after a long absence, Rich Condit.
Hi, Vincent. Hi, everybody.
How have you been?
I've been fantastic. This is all real good.
You were in Texas and then Spain, is that right?
I was in Texas. I missed a TWiV because I was in Texas, and then I was in Spain and missed two, plus I think there was a blank week. There was a TWiV blackout, is that right?
There was a TWiV blackout last week. Yeah, we weren't allowed to broadcast because it was a local game.
Because of the ferrets?
That's right. The ferrets, yeah. So you were in Buda, Texas.
I was in Buda, Texas. Wow, yeah. Boy, that seems like an age ago.
So I was just, I don't know, somewhere, and I start getting these Facebook messages. People are like, is that Rich in Buda?
Right.
And this great picture of you in downtown Buda, I guess, right?
Yeah.
That was cool.
And,
it was a good idea.
Who's our correspondent from there now?
Neva.
Neva, right.
Did you go visit Neva?
No, I didn't. I was kind of hoping she'd be there. You know, Buta's not real big.
I would think, yeah.
I kind of figured she'd be hanging around downtown by the sign or something like that, but she wouldn't know what I look like anyway. So.
You should have done a hangout. You should have tweeted, hey, I'm here in Buda. Come, let's talk about viruses. Right?
Good idea. At any rate, we will meet someday, Neva and I, okay, because Buda, is right in my daughter's back door.
Yeah, that's cool.
All right.
All right. Well, also joining us today from Western Massachusetts, where apparently the internet is now working, Alan Dove.
Good to be here.
It's nice to hear your clear, continuous voice.
Yeah. The connection has been up 5 days, 18 hours, 41 minutes at this point. I've got my router page open, and I just like to look at it occasionally and say, it's still working.
That's funny. That was a hellacious episode 2 weeks ago.
Yeah, and by the way, the only reason it went down 5 days ago was we had a brief power outage.
So the problem was your modem.
Yes, the problem was apparently the modem.
Physical problem. Well, that's good to know.
Yeah.
Well, good luck. I hope TWiV does not jinx you.
I hope not, too.
All right. Good to have you back. We also have an in-studio guest today, and this is actually someone that Rich knows very well because he is in Rich's lab at the University of Florida, Gainesville. Nissin Moussatche.
Hey, hello.
Did I pronounce your name well?
Yes, perfect.
Moussatche. You put a little A on the end, that's correct?
Yes, so depends on the side of the family, they put the A or they don't put it.
You know this guy, right, Rich?
I know him well. We met first in, what was it, Nissin? August of 1977.
September of 77.
77?
September of
Yes.
77, right?
Wow.
Yeah.
And we were together in Joe Cates' lab in
Stony Brook.
For about a year. Then we went our separate ways, though Nissin had his own gig in Rio, working on Pics that whole time. And then, as I like to say, I moved to Buffalo, and Nissin never showed up there.
Very cold.
The minute I moved to Florida, okay, he was on my doorstep.
I help you unpack, right?
Long story short, he's retired from his position in Rio and hangs out with me, and we have a good time together.
Long-time collaborators, right?
Yeah, it's a wonderful situation.
So you had a career in Rio as a professor working on pox viruses, right?
Yes.
You were born and raised in...
Born and raised in Rio de Janeiro,
Wow.
Ipanema.
Yes. Nice.
Yeah.
And then you retired and went to work in Rich's lab.
Yeah, I retired in 2008, but before that I was going back and forth because the family was already in Gainesville. And finally, Rich got money, and then I started working for him.
Nice.
Yes.
And then when Rich retires, I guess you will again. You'll have your second retirement, right?
Got my second retirement. Then I go back to the Ferdinando Beach.
So we should mention we did a paper of one of your students, right? Not too long.
Collaborator now, right?
Yeah.
Yes, ex-student, Clarissa Damaso.
She was your PhD student in Rio, right?
Yes, she was my PhD.
Nice. Actually, we have an email from her today. We get it all together here on episode 191. Well, welcome to the studio, and hope you enjoy.
Yes, I'm enjoying. This is really cool.
We will we'll talk about some things you might be interested in. Well, they are viruses, so you must love viruses.
Yep.
Being a virologist. We do have two cool stories today, and we do have, before we get to that, a follow-up from, was it last week's episode?
Yes.
Let me see.
Yeah, because this is about what's an aerosol.
That's
I found
right.
this a fascinating email. This is really good.
So this
Matter of
is,
fact, I want to communicate this to the medical students. This is really very
yeah,
good.
it's a good job. So this is from Russ, who writes, "Hi, Vince and the TWiV crew. I love the podcast. It keeps me in touch with lots of basic research in virology, which I would otherwise miss. I do pediatric infectious diseases, and you can imagine how much time we spend dealing with viruses. In my spare time, I do hospital infection control. I wanted to comment on your questions about this statement in the Fouchier paper. Quote, the pandemic and epidemic influenza viruses that have circulated in humans throughout the past century were all transmitted via the airborne route, in contrast to many other respiratory viruses that are exclusively transmitted via contact."
And when you guys read that statement, there was puzzlement over what that really means.
Right.
So Russ is filling us in, which is great.
Kathy and I couldn't figure it out, so we
And
asked for
Alan
help.
had dropped off of the internet.
Yes, Alan couldn't figure out how to take back the podcast.
So Russ, this is what's great about TWiV. Russ chips in. Here we go. In hospital infection control, we institute isolation precautions based on the usual route of transmission of the organism of concern. These are in addition to standard or universal precautions, which apply to all patients to prevent exposure to blood and body fluids. The guidelines come from the CDC and are, for the most part, evidence-based. They are airborne. These are organisms that can spread by small airborne droplet nuclei for distances of greater than 3 to 6 feet. The list is pretty short and includes tuberculosis, not a virus, varicella zoster virus, measles, smallpox, SARS, and avian influenza. It was also applied to the 2009 H1N1 influenza, but probably wasn't necessary. It is known that VZV can go under the door and infect someone down the hall. Interesting vision there. Droplet. These are spread by large droplet particles that spread no more than 3 to 4 feet after a cough or sneeze. Lots of organisms here, but the principal respiratory viruses that fall under this are seasonal influenza, rhinoviruses, and adenovirus. Also includes other infections such as mumps, diphtheria, rubella, pertussis, mycoplasma pneumonia, parvovirus B19, plague, group A streptococcus, and patients with meningitis caused by Haemophilus influenzae type B and Neisseria meningitis, where respiratory carriage is expected. And then the third one is contact, spread by hands and other forms of direct contact, stethoscopes. The main respiratory viruses here are respiratory syncytial virus, human metapneumovirus, and parainfluenza viruses. RSV, not Rous sarcoma virus, is a huge problem in children and also in the elderly and immunosuppressed. No vaccine yet, and there is a huge need. I've attached a quite old but instructive clinical study showing that RSV is spread by contact and not through the air, at least not as far as 6 feet. The setup is not dissimilar from the model for showing airborne transmission in the ferrets. In case you are wondering, folks with lice also get contact precautions. I guess lice don't jump.
No, they crawl.
So flu gets droplet precautions and RSV contact precautions. Go figure. Keep up with the great podcast. So Russ is a a doc in infectious diseases down at Tulane University in New Orleans. Thanks, Russ.
Excellent,
that's really
excellent
terrific.
summary. This is really good.
So the statement in the Fouché paper talks only about airborne and contact, and I was not aware of the fact that there is, from the hospital point of view, a distinction between airborne, droplet, and contact. And in fact, the statement that many other the statement that many other respiratory viruses are transmitted exclusively by contact is not untrue.
Mm-hmm.
But very interesting.
Yeah, I didn't know this division, so it's a different perspective, of course, right?
Did you look at this? I didn't.
Did I
This
send
paper
you?
showing the setup to show about RSV spread?
Did I not give you the link to
I
it?
didn't see that. No.
I don't see that in there.
I did put it online, but I forgot to give you guys the link, so let me...
I remember a talk once at a Gordon conference that was a human experiment about rhinovirus transmission.
Mm-hmm.
Okay, where they had people in rooms that were, it's like the ferret cages,
Yeah.
right? Where they
Sure.
they couldn't touch each other, okay, but they could communicate, and it was hilarious. It was so well done.
So how long a droplet can be infective in a situation like that?
Mm.
I don't know. I would guess that depends. That's another variable. Probably
That would be
depends on the
that would be very tough to set up a study for,
Yeah, that's certainly
because you'd have to track the droplets and see how long they've been sitting. I would think, though, I mean, given that they're talking about 3 to 4 feet after a cough or a sneeze, once it falls to the floor, it's probably not a whole lot of danger.
Mm-hmm.
Well, I cannot find it, but I will.
It's okay.
I'll post a link to it. It's a old. It's an old paper on a nice transmission study.
Cool.
I had meant to post it, but I forgot. But we'll do that. All right. Thanks for that, Russ. All right. Our first story today is something that has been in the news since around the 4th of July. It is an outbreak of some sort of disease in kids in Cambodia.
Yep.
And this is an outbreak that's actually been ongoing since about April, the beginning of April, and seems to have ended as of July 5th. According to WHO, 59 children have been affected. 52 have died. So this first was pointed out by ProMed Mail on July 4th, an unknown deadly illness in Cambodia, killing at least 60 children in the past months, and that number has been revised. And these kids go in, they have severe fever, respiratory and neurological symptoms, and most of them die. And at the time, they didn't know what was going on.
Most of them who come to medical attention die.
Yeah, right.
I'm sorry,
exactly.
The ones who come to hospital, you mean?
Right.
Yeah, and they come in really sick.
Yeah.
Right. So this is these are kids in Cambodia who have shown up to the hospital. Of the ones who show up to the hospital, 52 out of the 59 died.
Right. So they say in this time of year about 50 kids a day get admitted or show up to this particular hospital. It's a children's hospital.
Right.
And then these kids, they say they had encephalitis and they develop some kind of pneumonia, and then they die very quickly within 24 hours of being hospitalized. So that was the first ProMed Mail. And then subsequently, there's actually a whole list of ProMed Mail descriptions of this. Subsequently, a number of them, so they started doing lab tests, of course. Most of the kids were under 3 years old, by the way. And most of them, they didn't have lab samples because they died before they could take any. But early on, they found that quite a few of them were positive for enterovirus 71, which is a virus close to my heart because it's highly related to polio virus. It's a Picorna virus of the enterovirus genus. But not every one of these kids has been shown to have it, partly, of course, because, as I said, they die before they get samples. The kids are negative for H5N1, other flu viruses, SARS, and Nipah virus. And then apparently they have also found dengue and Streptococcus suis in a number of these kids. So the latest ProMed Mail post says that WHO is going to announce within a few days that a combination of Entro71 strep suis and dengue virus is the cause, in addition to inappropriate treatment with steroids. But that hasn't come out yet, so we don't have any more details on that.
Right, and it's not clear to me what inappropriate means, okay? Because I think that, for a lot of situations like this, steroids might be appropriate, but for this particular thing, it proved to be inappropriate.
Yeah.
Right, I think they were, it sounds to me like they were probably an appropriate choice given the lack of information.
Yeah.
You've got a kid dying of this whole respiratory immune response, and one medically appropriate way of dealing with a situation like that, if you don't know what's causing it, would be to suppress the immune system, because that's presumably a big part of the problem, and you do that with steroids. But in this case, if the WHO now has the data saying that it's EV71 with these two other pathogens ganging up, then suppressing the immune system is not going to be a good idea.
Right.
So, Vincent, it occurs to me that many of our listeners may not have this sort of global perspective on Picorna viruses. This might be an opportune time to do this, because you're talking about a fairly large family. They all have a very similar structure: positive sense RNA, naked capsid, but a really broad range of disease phenotypes, right?
Mm-hmm.
So maybe you could give us some perspective on that.
So the Picorna family is pretty diverse. It has all viruses with a number of things in common. They have a similar capsid. They have a similar plus-stranded RNA genome. And there are pathogens of people and animals as well in this family. And it's been growing as we isolate more and more viruses. And, of course, the most famous may be Polio Virus, which is in this enterovirus genus. And this is a virus that you ingest. It replicates in your intestine. It spreads to other tissues, in particular the CNS, and then you excrete it in the feces. Within the enteroviruses, there are also Coxsackie viruses, which can cause CNS disease, but also respiratory disease, muscle infections, skin infections, other echoviruses, which are similar as well. And then, of course, the rhinoviruses are also now classified as enteroviruses, even though they don't initiate infection in that particular part of us. And they are respiratory pathogens.
Common cold.
Common cold, right. And they're spread, as we heard from Russ. And what was that? Was that the aerosol? I forgot already. What did Russ say? Rhinoviruses are spread by droplets, no more than 3 to 4 feet after a cough, but also by touching as well. Then you have the famous foot and mouth disease virus, which is very different from the hand, foot, and mouth disease, which is the
and is
topic here.
Is that also an enterovirus?
No, that is an aphthovirus, a different genus.
A whaty virus?
Aphthovirus.
Aphtho, A-P-T-H-O?
Yes. And
okay.
these are pH-sensitive viruses and have other properties which distinguish them from enteros. There are actually quite a few genera now, much more than when I first started in this field.
Right. So under Picorna, you have entero, aphtho, and some others.
Yeah, and then you have cardioviruses, which are the animal pathogens like mangovirus and encephalomyocarditis virus, and then a whole range of others. Hepasiviruses, hepatitis A virus. Actually, Hepaci is a different family. Hepatovirus genus, that's the hepatitis A viruses, and a whole range of others.
Yeah, so I'm looking at Viral Zone here. Kobu virus,
yeah.
Perico virus.
Yep.
So, there's a ton of them. But in terms of human infections, it's mostly enterovirus, is that right?
Well, there's some new human picornas which are distinguished in different genera, but most of the infections are within the enteros. I would say the vast majority of human infections are caused by enteroviruses. And that may change in years, but the others are not as prevalent, yeah, for sure.
And even though they're called enteroviruses, it includes viruses that are transmitted by a fecal-oral route and viruses that are transmitted just via a respiratory route, correct?
Yeah, it includes the rhinos, yeah.
Okay.
Because they were originally given a different genus based on the pathogenesis, but when they were all sequenced, it made more sense to classify them together with the enteros. But the entero name stuck.
Right.
Which probably
So this hand, foot, and mouth disease, right, is initially a respiratory infection that's got fever associated with it, but then blistering around the mouth and on the soles of the feet and the palms of the hands. Is that right? Blistering lesions?
Yeah. You get these red spots, hand, foot, and mouth lesions, right? It starts with fevers and these sores developing in the mouth and other parts of the body.
Right, and it can be caused by a couple of different viruses, right?
Yeah, some of the Coxsackies cause it. A16 will cause it, and Entero71. And typically, it's very prevalent in Asia, and it's typically a benign illness. But sometimes the kids can get neurological complications, respiratory symptoms, and they can die. And so far this year in China alone, I think there have been 250 deaths of hand, foot, and mouth disease caused by EV71. So it's not unusual that the virus is lethal. I know a lot of the early coverage of the Cambodian outbreak after EV71 was identified, people were saying, oh, it's a new variant of the virus that's killing them. Well, not necessarily, because it's known in the past to kill kids.
Right, and that's one reason I brought up these are kids who actually showed up to the hospital.
Mm-hmm.
So depending on the setting, people don't take their kids to the hospital just for any old sneeze.
Right.
And it may be that there are a lot of kids out there who have hand, foot, and mouth and who are just developing the rash, or they get a little cough and parents don't make anything of it and it clears itself.
Yeah.
So by definition, we're looking at the most serious cases.
Yeah. There are a lot more infections of this virus than are lethal. There are many, many thousands and thousands of infections throughout Asia, and most of them are not lethal. And so there's something going on here that has to be sorted out. And whether it involves multiple pathogens, who knows, because we don't have any data yet.
Do you know if there's a genome sequence on these isolates, or whether it's one or several, or whether it's the same as stuff that's happened before? Is there any information on that at this point?
Not yet.
Okay.
I actually emailed Ian Lipkin last night, and he said he was trying to get samples, but there is a Pasteur Institute down in Cambodia, and they're working on this, and I presume they'll get sequenced pretty soon. It's easy to get these days.
So now hand, foot, and mouth disease also shows up in the U.S., right?
Yeah, it is. It's not terribly common, but you do find it in the U.S. I got a chart here.
And is that mostly Coxsackie?
Let's see. Other regions. EV71 is found in Africa, Europe, and the USA, causing sporadic outbreak. For example, during a one-year study in Canada in 98, 20 children had EV71 diagnosis. So it's not a lot. But, it's probably not diagnosed very often. Because, sometimes your kid gets a rash, and it goes away, and that's it, and you never do anything further. So the numbers are probably underestimates.
Now, in Asia, though, it seems to have this, you sent this review article, which I don't know if it's open access. Oh, it's The Lancet, so it's probably not.
It's not.
Unfortunately, it's a good review article. So this review on EV71 shows the outbreaks of this, the epidemics of this in Asia. I think it was in Malaysia specifically that they did this. And there's this periodicity. Every 3 years or so, you get this spike of infection, and I think the authors comment that it's probably it may be as simple as just that's how long it takes to get a critical mass of susceptible hosts.
Could be, yeah.
That now everybody under 3 is susceptible, and that's enough, and so you get another spike. And I noticed in looking at the graph, you've got 2003 was a big one, 2006, 2009. So here it is, 2012. And there's this epidemic of hand, foot, and mouth disease in Asia. It seems like we're kind of on schedule.
Could be. I suspect there are a lot of asymptomatic infections, and what you end up getting is immunization of the pediatric population.
Right.
Right. So even though you have X cases, there are many more kids who are infected. They become immune, and then there's no more host for the virus. Right.
Right. Maybe it circulates at some very low level for a few years and then breaks out again.
By the way, the virus was first identified in California in 1969.
So this virus is different from the foot-and-mouth disease from cattle?
Totally different virus, same family, Picorna,
family,
but different genus and very different virus, Some people mix them up because hand, foot, and mouth and foot and mouth disease. You have, they got the same components, right, except the hand, because cows don't have hands.
They don't have
Right. Well, and in fact, to put it in a perspective of an evolutionary tree, EV71 is, I guess, genetically closer to polio than it is to foot and mouth disease of cattle.
Yeah. So there have been recent outbreaks in Malaysia and other areas with substantial neurological components, where more kids have neurological complications than you would see elsewhere. And then you associated with that, you typically have death. So the virus clearly is replicating in the skin and in the mucous membranes of the mouth. That's what causes the rash. But it's obviously able to get into the CNS. So in that sense, it's polio-like.
Right.
So one of the suggestions I think I saw in some of this literature is that the polio vaccination or eradication might possibly impact on the epidemiology of this virus. Does that make any sense to you?
Not biologically, because there's no evidence that would happen, right? There's no evidence that polio has been keeping EV71 at bay all these years. Right? I think probably as polio goes away, then you focus your attention elsewhere, right?
Right.
And so in recent
we're going to
years,
notice this more.
We notice it more, but there's no biological evidence that polio excludes this virus or that there's any cross-immunity or anything like that.
All right.
Yeah. This review article suggests that these pulmonary symptoms that arise in serious cases could either be a consequence of neurological damage or cytokine storms, but it's not clear that there's any virus actually replicating in the lungs of these sick people.
Okay.
Now, the first author of this review, Tom Solomon, he's over in the UK. He's at the University of Liverpool, and I heard him talk in Dublin. Actually, he works on viral encephalitides, and I went up to him afterwards and I said, "Hey, why don't you come on TWiV and talk about this?" Because he's a clinician who is interested in all kinds of viruses that cause CNS infections or RNA viruses. It turns out he's going to be in New York in August, so he's going to come over and we'll have him on. And he can perhaps give us more insight into this as well.
Yeah.
Excellent.
Yeah, so one of the other things that came up is there's kind of this puzzle about why Asia.
Yeah.
And I guess people have floated a few ideas. One is that maybe it's an HLA haplotype that's more common in Asian populations that predisposes people to have a worse form of the virus, or maybe there's some co-occurring pathogen, which I guess would kind of line up with what the WHO is saying about this particular outbreak in Cambodia. But I gather that's just, it's not clear why that's happening, especially in Asia.
I mean, of course, it's spread by direct contact or by contaminated with stool, right? So it could also be a function of sanitation.
Right. Population density.
Yes. So we don't really know. The HLA is a good idea, but there's no evidence as far as I know.
Right.
And we should point out there's no vaccine and there's no antiviral available. So knowing, as someone had, I think on the WHO report, or maybe it's a ProMed mail, it doesn't matter that we've identified EV71 because it doesn't help us because we can't treat it. Yeah, the link to EV71 does not particularly help in the treatment of the illness, as there's no antiviral and no vaccine.
Well, it does help tell you that maybe you shouldn't give steroids.
Yeah, that's for sure.
I mean, that sounds like progress of some kind.
Yeah.
And also this, not to harp on this periodicity of it, but it gives you some ability to predict that this type of thing might be rolling around again in a few years.
Yeah.
So you've got to.
So, I don't know what the role of dengue and strep suis. Strep suis is a pig pathogen, which often can spread to people. It's not terribly common. So I don't know why they're saying, we don't have the data, so we have to just wait and revisit this to see what's going on. At the end of one of these ProMed articles, it says, it remains probable that EV71 is the major component in this outbreak. The involvement of other pathogens can't be discounted. But we don't have the data, so.
Yeah, and it's going to be tough because 60 is not a large number. Okay,
No.
so
Right, and they've all died, and if they don't have samples, that's it, right? Not much you can do.
It might help to go into the countryside and see if you can get at least serosurvey data and see how big this thing is.
Yeah.
I'll be very interested to see. I'm sure that we will see genome sequencing out the other end and see if this is one or multiple strains and if they're the same or different than strains that have been around before.
Right.
I think it really would be nice if you could do a human genome sequence really quickly on some of these patients, right?
Someday you'll be able to do that.
Yeah,
Yes.
someday, because then you could see if there's some predisposing mutation. We've talked about some of these mutations on TWiV in the host that make you susceptible to serious disease, right?
Mm-hmm.
Could be a combination of things.
Yeah.
So it's really interesting. The general mindset is still to blame the virus, right? It's a more virulent form. But, when people get sick and go to the hospital, you have a selection process right there.
Mm-hmm.
Yeah.
And those could be people with gene alterations. Anyway, I guess we'll see this again in a couple of weeks. All right. The next story is a paper that was published on Thursday in Science, and its title is Attenuated Vaccines Can Recombine to Form Virulent Field Viruses. And this is a one-page paper. I guess it's a brevia. Yes, it is.
Yeah, it's one of these cool brevia.
Brevia work for me. This is great.
I love these. It's about right for my attention span.
Yeah, exactly.
Bang, bang.
You know?
There's the paper. Here are the data. And then you're out.
Spare me the details. Just
Listeners, these guys can read hundreds and hundreds of pages. Don't let them say that. They just like it when it's shorter. It's very short because it has most of the third column is references and acknowledgments and other stuff.
And really, figure 1 is the money shot. I mean,
It
you
is.
pretty much know what's going on just by looking at this figure.
It is. This deals with a virus that infects poultry. And, everyone talks about avian flu, but there are other viruses that infect poultry. In fact, it's a herpes virus, Gallid herpesvirus 1, or infectious laryngotracheitis virus. Now, Alan, Gallid herpesvirus 2 is also known as Marek's disease virus, which I know you know about.
I have heard of that.
Because you've interviewed the man who made the first vaccine against it.
Yes.
Maurice, right?
Yes, that's right.
So that's a different virus. That's actually an oncogenic herpes virus. But Gallid herpesvirus 1 can wreak havoc in poultry worldwide. And, so you want to not have this disease if you're raising chickens, for example.
Yep.
You don't get meat, you don't get eggs, etc. So there are vaccines that are used. And it's typical in veterinary vaccines. There's more than one because I think the energy barrier for making a vet vaccine is much lower than for a human vaccine.
Well, compared to human vaccines, it's almost non-existent.
Yeah.
Because what we would consider a preclinical vaccine for humans, which is very, very early, you don't even put money into that because it's probably not going to pan out. In veterinary vaccines, that's put it in a bottle and sell it.
Yep.
Because animal testing is as far as you need to go because you're going to be giving it to animals.
Yeah. So if virologists out there in training, if you want to see your work go to practice right away, go into the veterinary or agricultural field.
Yeah.
So there are typically multiple vaccines, and so that's the case here. In this particular study, it was done in Australia, where they used 3 different kinds of vaccine against Gallid herpesvirus 1. And they're all what we call live attenuated viral vaccines. So they are given to the birds and they replicate. And so there's one called the Cerva vaccine, which is of European origin. And then there are two Australian origin vaccines. They're called SA2 and A20.
Both closely related, right?
Highly related. And those are different from the Cerva vaccine.
Right. So as attenuated vaccines, these have mutations that prevent them from being pathogenic.
And we probably should say that we don't really know what the mutations are. They're just passed in various ways until you empirically get a virus that doesn't cause disease in a chicken.
Right.
And it's easy to do those experiments because you don't have to have a human trial license. It was called an IRB approval.
And by the way, I mean, it's going to come up repeatedly as we discuss this, the sort of relative sloppiness of vaccine development in animals. Personally, I wouldn't argue that there's any need for more regulation of that or anything, because these are vaccines that are going to be given to millions of birds that are worth $1 or $2 a piece or whatever, and you wouldn't want the overhead of the vaccine cost of a more elaborate development process.
Sure. Yeah.
Not only that, but you're going to slaughter the birds in the end anyway.
Exactly. These are birds. They're going to be around for a few months, and then you're going to wipe them out, and nobody cares about that. So why you're not going to be?
I don't want to be callous about it, but these are animals that are being raised for slaughter anyway.
Yes, exactly.
I'm sure there are people who are going to take issue with that, but
I'm sure.
You're right.
No, it's an observation of fact. I understand there are vegetarians who object to the existence of that process, but given that that's the way we raise our meat, we need economic ways to prevent disease in those animals.
Just an aside, the rise of growing animals for food, chickens, pigs, what else? Cows, I suppose. You put them all together, crowded, and you get these outbreaks of disease, which normally wouldn't happen if they're just roaming around, right?
And would not be nearly as big a problem if you weren't doing this on such an industrial scale.
Yeah, right.
But
exactly. But then you'd have to go out and trap them, which would be...
Or you'd have to do a bunch of small farms, which would have a bigger environmental impact anyway.
Yeah.
I'm reminded of shows we've done on aquaculture. Where you stick a bunch of fish together.
Yeah, right. And, same thing, exactly. Yeah.
Same thing.
Get infections. All right, so they used 3 vaccines in Australia, and starting in 2008, there was new viruses arising, causing disease in chicken farms.
After they'd introduced a new vaccine.
Yeah, they had introduced the Cerva vaccine in 2006, right? So these viruses are distinguished according to genotype. I suppose they sequence them or look at polymorphisms in the sequence and they classify them. So the new viruses that emerged were class 8 and 9 genotype viruses. The Cerva vaccine is a class 7 and the other 2 vaccines are class 1 viruses. So basically what they did in this paper is they sequenced all of these viral genomes. They sequenced the field isolates, the class 8 and class 9 field isolates, and they sequenced the A20 and the SA2. I think the Cerva sequence had been done before, and they just aligned them, and boom.
They aligned.
They align, and you can see that the new field isolates are derived by recombination of Cerva and one of the other 2 vaccine strains. So the class 9 comes from Cerva and A20, and the class 8 comes from Cerva and one of the 2. They can't really tell because the sequences are so similar.
Right.
It's quite clear from this figure. You can see
absolutely
the crossover.
Absolutely clear. So you take 2 related but attenuated viruses that have different attenuating mutations in them, which means that they have also a lot of normal virulence genes that are still intact, and you mix them together and they recombine with each other, and I guess really the animals select out of that viruses that have retained the virulence genes, because those viruses grow well and they emerge.
Yeah, I think that's the selection, right? The growth.
Yeah.
And that turns out that happens to be virulent as well.
Yeah.
Right.
And they take, so they do a number of other things. They take these isolates and they put them back in chickens and show that, in fact, they are virulent. So it looks very much like that's what happened. These things recombined in the field and restored the virulence. And as Rich said, the problem is that they're different to begin with, so recombination can restore a more virulent virus or a replication-competent virus. Let's see, anything else here? Yes, that's about it. So they say at the end, these findings have implications for the use of herpesviruses and possibly other DNA viruses as attenuated vaccines or vaccine vectors.
Yep.
So the question is, let's talk about humans. Is there any chance that this could be an issue with human vaccines?
Not currently.
Why not?
Because of the human vaccines that we give, we don't have any that would have that would be given, in a way that would allow this type of recombination event.
Mm-hmm.
I mean, to the extent that we have combined vaccines, there are things like, first of all, it would only apply to the live vaccines. The killed vaccines are not going to recombine.
So the live vaccines
are what? Measles, mumps, rubella.
Right.
Flu mist.
Right.
Chicken pox.
Chicken pox.
Is, in fact, an attenuated herpes.
Right.
That's the only one that's DNA virus, right? All the other ones are RNA virus.
Well, there's an adeno vaccine, but that's just given to the military, although those a lot of people, I guess.
Right.
There's the papillomavirus vaccine, but that's a virus-like particle. That doesn't have any DNA
in it.
So
and there's the hepatitis B vaccine, but that also is just a subunit vaccine.
Right.
The rotavirus vaccines are infectious. And the polio vaccines.
Right. But we don't have... I think the one that would be the best candidate for this would be the polio vaccines.
But the polio is the Sabin, not the
Sabin,
right,
right.
the Sabin, the live attenuated that we don't give in this country anymore. But that's still being used a lot worldwide.
Yeah.
There you actually do have 3 strains that could, in theory, exchange genetic material.
Except that they revert long before.
Exactly, and in probably everybody who gets those vaccines, you get revertants anyway.
But can RNA virus do recombination, recombine?
They can, yeah. It's been
Some
shown.
yes.
Yeah.
It's been shown with polio, and in fact, it turns out that when you get the polio vaccine, the Sabin vaccine, not only does it revert within a few days, but what you excrete are recombinants among all 3 serotypes.
Yeah. But that's reversion, not recombination.
No, you get both.
Oh, they get both.
You get enormous recombination in your intestine. So what you get out are multiple crossover viruses. It's very interesting. And in addition, if someone gets vaccine-associated polio after taking the Sabin virus, almost every one of those vaccine-associated isolates are recombinants between the Sabin virus and a non-polio enterovirus that happens to be in that person. Okay, a non-polio entero. And they can tell that by sequencing it. So all of them are recombinants.
Yeah.
And those recombinations remove the attenuating mutations. So I think the polio vaccine is a good example of this already. We know this happens for the polio vaccine.
It does actually happen.
Yeah, it does.
But not commonly enough for it to justify not doing the vaccine.
No, absolutely. I mean,
and it's actually something we've known about for a long time that's a risk of the Sabin vaccine.
So the risk is about 1 in 1.5 million doses, and that's why in the U.S. we stopped using it because there was no more wild polio and it could no longer be justified. But
actually, I'm very interested in those references, Vincent.
Yeah, I can send them along
for the polio stuff. So,
and for go ahead.
Go ahead. No, you go ahead.
No, for the global eradication campaign, just to kind of tie that up, they've now shifted to using a monovalent vaccine for the most part, right?
Yeah, monovalent 1 and 3. The 2 is pretty much considered to be gone,
right? And
so they're doing monovalent. So yeah, you would still get recombinants with the non-polio enteroviruses in those cases. You wouldn't get the recombinants among the vaccine strains.
But again, it's a well-characterized risk.
Are we talking about a variety of different enteroviruses, or is there some predominant ones that cause this problem?
Well, they are mostly nothing that we know about. You pick up the sequence, and it's clearly an enterovirus. It's not polio.
You haven't seen it before.
You haven't seen it before, and it's typically it's typically something new. And they're different. They're all different. Not a huge number have been sequenced, but they're pretty much all different. Now, I don't think that. I mean, at the same time, reversion also plays into this. You get a lot of reversion, so it's one or the other. You're going to get recombination or reversion. As long as you're using the live polio vaccine, there's not much you can do about that.
Right.
But that's the only one I think would be an issue. I looked into the AD vaccine, and it is a mixture of two serotypes, AD4 and 7. It's a live attenuated vaccine. I assume they were made independently, so they probably have different mutations, right?
I would assume so.
I would assume so, and I think they could probably recombine and make a wild type AD4 or 7, but that would cause respiratory or GI illness, and I don't think that's any more serious than just getting adenovirus infection to begin with. So in terms of human safety, I don't think that would be a big issue.
Right, and the other live vaccines are things like MMR. You've got 3 viruses that are not they can't recombine with each other.
Right, they can't recombine. That's right. They're different viruses. Now, right. Rich, the pox vaccines used, are they different?
Let's see. Yeah. I mean, MVA was attenuated by passage in eggs and has all sorts of problems with it. That's a vaccinia that was passaged. See, okay. So let's put it this way, none of the pox vaccines start with smallpox and attenuate it.
Right.
Okay. They all start with what were initially naturally occurring viruses that are related to smallpox, but distantly enough so that they have never and probably would never cause a smallpox-like disease. Now, there are several different vaccines that have been derived from those viruses that are derived in different ways, largely by passage under different conditions that could theoretically recombine with each other to regenerate something like the original parental viruses, but those parental viruses are not really pathogenic to any great extent in humans anyway. Okay, so I don't see a problem with that.
Okay, so really, among human vaccines existing today, the only issue is really polio, which is known to recombine already.
Chickenpox cannot recombine?
I think there's only one vaccine, right? I mean,
that's worth looking into.
It's a chickenpox and a varicella zoster, but they probably are different.
And they're live, right?
They're both live, yeah. But they're given to different age groups, right?
Right. So the chickenpox and the shingles vaccine are the same vaccine.
Okay.
And the shingles vaccine is just given to older people and at something like twice the dose.
Right.
Okay, but it's worth, as far as I know, there is only one strain that is used as the chickenpox vaccine. However, that'd be worth looking into, and this is a sort of a wake-up call for that kind of thing.
Yeah. Yeah, you might not want to go developing additional herpes virus-based live vaccines. I don't know that anybody's doing that, but
right. Yeah.
This is a potential concern for future vaccine development.
Or for gene therapy, it's the same thing.
Yes.
You want to be using the same backbone, right?
Yes.
But that's not likely to happen if different companies are trying to target the same disease, right?
Well, you'd have to get an odd set of circumstances to have something like this happen, though. Because the ways different companies are using to target the same disease are usually things like, somebody's using an adenovirus, somebody else is using a DNA-based, pure DNA-based approach. So they're radically different in most cases.
And these vectors tend to be gutted, right?
Right.
Like the gutless vectors. They really cannot recombine to form a parental virus.
And the patent attorneys kind of ensure that the approaches are going to be radically different, frankly.
So in this Australian herpes virus of chickens, my impression is that the problem is that they use multiple vaccines to immunize the same flock, right?
Right.
And that's where you get the same virus in both viruses in neighboring chickens, and they can spread and recombine. But people typically are not going to get two different vaccines or two different gene therapy modules, right?
Right. That's the other thing with gene therapy. It's targeted, at least currently, it's targeted to very, very narrowly defined patient populations.
Mm-hmm. So we all agree then that the risk for humans of this happening is probably very, very low, or being a problem?
I would agree with that.
Yeah.
Mm-hmm.
Yeah.
I would say, however, that this is an important finding because it raises awareness of the notion that this sort of thing could be an issue, and that people ought to think about it as they develop vaccines or vector-based gene therapy stuff. And in fact, I've told you guys before that I am a member of this committee that's a subcommittee of the Brighton Collaboration that is trying to develop international guidelines and standards for the use of viral-based vectors. And one of the things that they're working on is guidelines that deal with this very issue, as mandated by the World Health Organization. So at the World Health Organization is tuned into the notion that recombination amongst viral vectors or vaccines could create an issue, and they want people to think about this. And so this Brighton group is one of probably a number of different entities that are considering this problem and will come up with recommendations. So it's something that there is an awareness of, and I think that this is an important veterinary example that highlights that that it is something we should be aware of, and developers and manufacturers need to take this into account as they develop new vaccines.
Yeah, this is not something that just came out of left field.
Right.
This is a concern that people in vaccinology have talked about for a long time. Yeah, what if? But what's cool about this paper, or interesting about this paper, is it's actually happened. This is the first empirical demonstration that it's not just a theoretical risk.
And I'm sure that Brighton will be interested in this paper, right, Rich?
Absolutely. I sent it to Bob Chen the minute I got it. Well, the minute it was published.
That's right. There were two things I wanted to mention I skipped over from this paper. First is that they went into the field and looked at more field isolates and found that they also were recombinants. And they didn't sequence those. They just look at restriction digests to prove that, because you want to go back out and say, is it just these two isolates that we have, or is it many?
Right.
So they looked at addition, and they were also crossovers in the same way. And then they took a couple of these isolates and put them in pathogen-free chickens and confirmed that they in fact had increased virulence.
Yes.
Right. So those are two important parts of this
Now,
study.
did they say whether these different isolates are all identical? I'm interested if these, because there's probably a gazillion different ways that you could recombine the vaccine strains to come up with a virulent isolate. But I'm wondering if these are basically clonal emergences, or whether there are a bunch of different strains.
Well, what they say is that the restriction fragment length polymorphism patterns were consistent with these two viruses that they looked at. And then they say PCRs revealed that the same recombinant genomes were present in other isolates.
Okay.
So the same sounds like identical, but unless you sequence it, I don't know, right?
Right. They haven't sequenced them all, and that would be a way to determine if this is something that arose once or if it arose multiple independent times.
Right. Today I actually received an email from Natasha, who wrote, I read your blog and look forward to all new posts. I was wondering if you were going to cover the news story on poultry vaccine strains deadly recombinant.
Timely.
Yes. And she writes, if so, I would appreciate if you could address the following questions in your blog, as none of the articles so far seem to be covering this issue. 1. How many genomes of viruses carried by people who have died or have suffered long-term effects following vaccinations have been fully sequenced? Okay. So the only data I know in really well is the polio data. So,
and that's quite a few.
It's quite a few. So people who have had vaccine-associated polio, or when there have been outbreaks of polio caused by vaccine viruses like the Dominican Republic and many others, these have been sequenced. We know that they are recombinants between the vaccines, among the vaccines, and also with non-polio enteroviruses. And we know that people get polio from these vaccines because they are virulent, but there is something different with those people versus everyone else who don't get ill, but they still have the same recombinants and revertants within them.
Right.
It is a patient issue that we don't understand. So, the vast majority of people who get these polio vaccines are fine. It's just these individuals who are not. So it's not a vaccine problem. It is a problem with the host, and we don't know what it is. Do you guys know any other examples of where vaccines have caused issues and they've been sequenced? I don't know of any others.
No.
But
my guess is, I'm not sure about this, but my guess is that in the case of the yellow fever vaccine, there has been some of this done.
Yes, that's
because
correct.
There's been some adverse effects that have come up. In particular, there's an increased awareness of some adverse effects that we were not aware of before with the yellow fever vaccine. And I think there's there's investigations going on as to what those are about, and I can't believe that those investigations don't include sequencing the genomes of the viruses that came out of those people. But so I don't know for sure, but that's a candidate.
How about someone who gets a vaccinia-based vaccine or pox vaccine, and they have rash or some different effect? Does anyone sequence those and see what's going on?
Not to my knowledge. Nissin?
No. I don't think they sequenced the kid, the little kid that was infected from the father. You remember?
Right.
Yeah.
Yeah.
Or even the people from the gym that were also have the son.
Oh, right.
That's right. And I'm not sure. Well, I guess those people are getting a canvas as a. I was going to say that in some of those cases it's not really informative anyway, because the old Dryvax vaccine was fairly heterogeneous anyway.
Right.
Right.
But the new vaccine, a canvas is a cloned vaccine. So if there were any variation from the parental strain, you could tell by sequencing. But as far as I know, that has not been done, but it's a good question. Once again, it wouldn't surprise me if because obviously it's the capability exists. If people had done that, I would have to ask. I would have to ask. Certainly, if it's been done, it's not been published to my knowledge.
Rich, you remember this case where we talked of the military guy who got the vaccine and then transmitted it to his girlfriend or wife or something?
Yes.
And she got some kind of infection as a consequence, right?
Mm-hmm.
So they didn't sequence that either.
Not to my knowledge.
Yeah. There's no indication that any reversion had to occur to
No, I mean in most of these cases where there's a problem with vaccinia, it involves some sort of immune suppression or something like that.
I think the baby was immunosuppressed. Or have like eczema.
Yes.
Yes.
I think
the baby has eczema, and then they have spread all over.
Right.
I think there was something in the military case.
There was
that was the military. The father was a military that was playing with the kid before.
That's the kid.
Yeah, being deployed, and the kid had all this all over.
The case I'm thinking about is another military guy. He spread it to his girlfriend, and she got a genital infection.
Right. But that was a localized infection. That was not a real complication. That was just a very common situation because there's live virus in the vaccine, and it causes these lesions. And one of the precautions that you have to take is not to spread it around. So that didn't involve anything really unusual. There was one other military case where a guy was vaccinated, and it was subsequently discovered that he had leukemia.
Mm-hmm.
And I forget whether he had started therapy or not, but basically either the leukemia or the therapy left him immunosuppressed, and that triggered replication of or allowed replication of the vaccine virus, and he got really sick.
Mm-hmm.
But once again, that's a situation where the host is immunocompromised.
Right.
So actually, this discussion addresses her second question, which is, following on the above, assuming the numerical answer is low or close to zero, how can we rule out with absolute certainty the possibility that some of the severe or prolonged negative reactions to vaccines are not due to recombinant or mutant viral strains, including the possibility of vaccine and wild strain recombinants. Are there any large-scale studies in the pipeline meant to address this question?
Right. So the answer is not low or close to zero for polio.
Right. Yeah, we know a lot about that, yeah.
And in terms of can we rule out with absolute certainty, well, we can rule in with absolute certainty that this does happen with polio. And it's a well-characterized risk of it, or as close to absolute certainty as we can possibly get. I don't think we can rule in or out anything with absolute certainty ever. Welcome to life.
Yeah, but we can, as far as with other vaccines, there's no evidence that this is playing into any adverse effects.
And at the same time, it is something that is on people's radar.
Yeah, it is.
And I think that it's a bigger blip on the radar now with this paper. Certainly on my radar, yeah.
No, it's something people have actually thought about and have looked at, and we've got one vaccine, the Sabin vaccine for polio, that we know does this, and we have, I think, very sound reasons to believe that this is not happening, and sound data to believe that this is not happening with any of the other vaccines that are routinely given to civilians, at least.
Right. And remember, by adverse effects, we're not talking about autism, because we know vaccines do not.
That's not one.
Cause autism. All right. It's other things like polio, vaccine-associated polio, or the yellow fever, vaccine-associated complications, that sort of thing.
Yeah, when you talk about severe or prolonged negative reactions to vaccines, you're already talking about an extremely small set of cases.
Right.
And the other thing is that this is
things like polio. Sorry.
It's all right. This is the bottom line here is risk versus benefit.
Yeah,
right?
Yes, sure.
We're talking about small numbers of adverse effects. Sometimes those sometimes we can do something about that. As you already pointed out, Vincent, we no longer use the live polio vaccines in this country because we can afford to use the inactivated vaccine and administer that. And one of the reasons that we don't use the live vaccine anymore is because of problems like this. And sometimes you could do that, sometimes you can't.
A reason that we don't give yellow fever vaccine to everybody in childhood.
Right.
You know that your risk of getting it is probably nil if you're not traveling outside the country, so you don't get it.
Right.
Now, I understand the yellow fever backbone is being used for carrying other antigens.
Correct.
Right, and there's no evidence that that's been an issue either. West Nile is licensed for horses.
Yes, as a matter of fact, in the development, that's a really good example of this issue of recombination. Potential recombination with wild-type strains and amongst vaccine strains came up during the development of these Chimerivax vaccines. And so they did a lot of very rigorous experiments to test whether or not recombination was going to be a problem in these, and all of those experiments proved negative. In this case, those viruses don't recombine very well with each other to start with. So they had to really force recombination to test for anything, and basically they couldn't. They came to the conclusion that there was not going to be an issue with this, and they did some very rigorous tests on these.
Right, and in that case, that was done because it impacts on a human vaccine.
Mm-hmm.
So, getting back to that, why this happened in chickens, you don't pay as much attention to vaccine development in chickens. But if you're gonna be messing around with something that could happen in a human vaccine, then the FDA is going to be all over you.
There's a nice summary of this article by Ed Young over at his blog on Discover Magazine, and he quotes the author, one of the authors of this paper, as saying using more than one live vaccine at the same time is a feature of veterinary medicine rather than human medicine.
Yes.
And that's pretty much what we have said. The title of his post is wonderful. Chicken vaccines merged to form live viruses and caused outbreaks of irony.
Yes.
That's wonderful. Thank you, Ed. We got a bunch of links here. We'll put them all up so you can check this out. And that will do it for our two stories today.
Great stories.
Yeah.
They're cool.
Yeah, very cool.
Virology is a great area. Always something cool. Should we do some email?
Sure.
Larissa. Hey, someone take this for me. Rich, can you take this?
Dear TWiV panel, I have been looking for a list of papers reviewed by the 3 podcasts recently, and I can't seem to find one. Is there a list on your website of papers reviewed, or should I just listen to TWiV, TWIP, TWIM with a pen and paper at hand always? Thanks for all the great dissemination of scientific literature. It helps fill what would otherwise be many unscientific hours. Warm regards, Larissa.
Well, we do have show notes for each episode, right? And you can find those at twiv.tv or microbeworld.org/twip or twim. And in the show notes, we always have links to the papers. I don't have a separate list. It's just another thing for me to do, which, I make a list. I make a list of weekly picks, and I make a list of, I archive all the letters. So to put another list, I think would be...
Yeah, plus I think the papers really need to be in the context of an episode.
Yeah.
So a list is not as informative as the other lists that you post.
But Larissa, you can find all the links in each episode show notes, right?
And you can also search the site if you're interested in a particular topic.
Yeah.
And that's pretty effective.
That'll work. Alan, can you take the next one?
Sure. Oren writes, Dear Lords of the TWiV.
Nice.
Oh, yeah.
I like that one. It is a pleasure writing to you after being a longtime fan. The weather in Tel Aviv is extremely hot and humid from April to October. The show is great and keeps me up to date in aspects of virology not in my direct field. Well, I probably got the TWiV bump from TWiV 115, Color Me Infected, as I am now starting my own lab in the medical school at Tel Aviv University. We're currently trying to upgrade the virology lab for the medical students and make it more appealing. We have a low-budget wet lab that complements the medical students' virology course. If you or your listeners have any idea for an interesting way to attract medical students, it would be highly appreciated. Keep up the good work, and thank you. And Oren is at the Sackler School of Medicine of Tel Aviv University.
So Oren did the 7-color herpesvirus work paper in Lynn Enquist's lab.
Right.
You guys remember that?
Yeah.
Yeah.
That was a great TWiV.
It was fun.
That was a great paper.
So any ideas for medical student labs? We don't do them here anymore, actually. We cut them out.
Yeah, we don't do them here either. They're all any labs are virtual labs.
Did you do them in Brazil at all?
Yes, we had some, not virology, but for medical students we have like isolating DNA and doing some other stuff. Yeah.
Well, maybe some listeners have ideas. We used to have them. Here's what we had them do here. We would have them do a bacteriophage plaque assay, which they didn't like. And then we would have them bleed each other and look in their serum for antibodies to polio. So
they probably liked bleeding each other. Right?
They like bleeding each other. And then we would give them some Sabin virus, so it's attenuated, and I often prepared it for them. And then they would do a neutralization assay, and they should all have antibodies, and the ones that didn't, you could immunize them. So that's a cool thing. It's not too hard. It involves them bleeding, which is, they're medical students. They want to stick needles into each other. Right? So that would be my suggestion.
Would that involve all sorts of regulatory hassles nowadays?
I bet nowadays that would be.
Yeah.
Well, can't medical students do that? They are docs, right?
Well, I think they go
Not
around
yet.
bleeding each other.
Not yet. Yeah, you're right. That's a good point. Maybe that's why we don't do it anymore, because of the IRB. I don't know.
Yeah, you got people bleeding each other. You got polio. You got all kinds of issues.
Well, you don't know if one of them could be like infected with some kind of virus that will.
Sure, yeah. I'm telling you, when we did this, it's the early 80s,
Wait, so if medical students aren't sticking each other anymore, how do they learn? Oh, I don't want to know.
Yeah, right.
Actually, I think they do stick each other.
Yeah, I'm pretty sure. My wife had to do
We're
that.
going to get mail about this.
Yep. My wife finished medical school in 2000, and she definitely was sticking her friends. And
Did she do a virology lab?
no, I don't think so. She went to Columbia.
Yeah, I remember us. That's right. Well, that's where you met her, right?
No, I met her at a viral pathogenesis conference at Mount Sinai.
How romantic!
but you-
I told this story on TWiV.
I must have missed it because I wouldn't forget this.
It was actually during a talk that David Baltimore was giving. So I was there. I had a poster there. I was presenting my my thesis work essentially, and this is 1995, and they, I was in this talk, or went into this talk after a break, and this cute chick sat down next to me just by happenstance, and she and her girlfriend started doodling little stuff to each other, and they were doodling visual puns of restriction enzymes.
Okay.
So I took my
puns are right up your alley.
I took my
Really?
paper, I drew a couple of little round donut-shaped things, and I handed it over, and this cute chick next to me looked at it and shrugged her shoulders, and I wrote Bagel 2 underneath it. That's it.
Man, the rest is
And
history.
that was the rest is history.
That's
Right?
it.
I want to know about the conference, the seminar that someone was talking. Do you remember? No.
No,
Doesn't remember the talk. No.
No, he doesn't remember the talk. Right? Okay.
I'm sure it was absolutely fascinating, but I really couldn't tell you what any of that talk was
Do you
about.
think, Alan? Do you think if she had not gone to Columbia, it might have ended there?
I don't know if she.
Because
I
it
don't-
was easier seeing her here, right?
Oh yeah, absolutely. I mean, I was already living in the neighborhood, and then she started at Columbia. She was a tech at Mount Sinai when we met, and then she was applying to medical school. She got into Columbia. So I don't know.
Her father was a well-known internist at Mount Sinai.
Yes, and her grandfather was a pediatrician.
So when I was a graduate student at Sinai in the 70s, I think her grandfather was still working there. His first name was Horace.
Horace Hodes, yeah.
Is that his name? Yeah, yeah. So he was a pediatrician?
Yeah,
I think he did ID stuff because he was teaching in the micro course.
pediatric ID, and then his son, David, followed in the family tradition.
That's
and,
right. David Hodes. I know. Was he at Sinai?
Yeah, he was at Sinai till a few years before he retired, and then he went to work at Roche for a little while.
Cool. That's neat. Now, why did we get on that? I forgot.
We're talking about medical students.
Oh, medical students.
Medical
and
students, yeah.
Yeah. All right. Anybody has other experiments for Orin, let us know. Next one is from Marcin, who writes, "Hello, virocides. Can you please provide your comments on the attached editorial? It seems rather depressing that there are investigators out there that believe that if scientists don't work 24/7, then they are not doing a good job. It seems that it is especially true at Johns Hopkins." So he sent a letter. He sent a link to an article entitled "Where's the Passion?" It was published in Cancer Biology and Therapy. It's by Scott Kern, and it's basically this fellow who is complaining that nobody's around on weekends in the place where he works, and he says that shows that nobody is passionate about it. Well, in my view, it doesn't matter how long or how often you work; it's what you do, and the passion that you supply to it. So I have seen people who do 9 to 5 in science, and they are brilliant and they are very productive. So you don't have to be here all the time. In fact, sometimes people are here all the time and they're very inefficient. So personally, I don't see why it matters as long as you do the work. What do you guys think?
I agree completely. This has been a thorn in my side for my whole career because I had my nose rubbed in this idea of 24/7 is what you have to do very early in my career. And it's not the way I wanted to work, in particular because I wanted to raise a family. And so I've been sort of fighting with this in my head my whole life. And I have to say that I've always worked essentially a 40-hour week. And while I'm not in the National Academy and haven't won any great prizes and et cetera, I've still got food on the table. And my lab is still functioning. So I'm a living example of the fact that you don't have to work 24/7 to be successful. You do have to, put in an effort, and it helps to be, in my view, it helps to be very organized. And to be able to multitask and to focus and make efficient use of your time. But you don't have to be there. There's some sort of weird machismo thing that goes on here about being there at weird hours and stuff like that really makes me kind of sick.
I think you hit it on the head with machismo. This is also something that has come up repeatedly in conversations about why there's this gender skew in science. And it's not that one gender or the other is better or worse at lab work. It's just there are more men who are willing to buy into this, what I think is nonsense about, you got to work all the time. And certainly you can point to people historically who've been successful scientists who have had 7-day work weeks. You mentioned Maurice Hilleman earlier. He was legendary for just always being there at work. But you can also point to people who didn't and who were just, arguably just as successful. I think, wasn't it Alfred Hershey? Who used to? It was one of those old guys. Used to set up his experiments in the morning and then go sailing.
Well, there's a good idea.
Work in Cold Spring Harbor, that's possible.
Yeah, I think it was Hershey who did that. Anyway,
smart.
but there are examples. And of course, Carrie Mullis is a legendary example of the Nobel Prize approach. I'm not sure that that's. Either of those extremes is really typical, but the point is, exactly as Rich said, it's like any professional position. You do need to put in an effort, and you do need to be smart and organized about how you work. But this idea that everybody should be there every single day is not only wrong. I think it's actively alienating to a lot of people who should have gone into science.
Mm-hmm. I agree.
You remember this? I think we had a reader send in a story a while ago about a PI who does this, and he works 24/7, and he only takes people who do that in his lab.
That's this other link you've got down here.
Oh, that's right.
Because I had seen this before.
Yeah, I think we talked about it.
Yeah, we've had this discussion before, but it doesn't hurt to have it again multiple times.
Yeah, that's right. This is this lab at Hopkins. Alfredo Quinones Hinoso. Yeah, that's right. He just works all the time.
And, if that's what he wants to do, and if that's what the people who work with him want to do, fine.
He was a 19-year-old illegal immigrant from Mexico, labored in the fields in California, and now is a neurosurgeon at Hopkins. He did not get there by working 9 to 5. I see that's... You don't have to. If you're really good, you don't have to work beyond 9 to 5. You could be great, right? Now, in his case, it worked, but that doesn't mean everyone has to do this.
Mm-hmm.
Right.
Yeah, this came up before. Yeah, we're going to put a link to this in again. That's right. This really bugged me back then because, as we have said, it makes everyone think that the key to good science is just to work all the time, and that's just not right.
And it's going to burn people out who are not, if that's your bag, if that's your way of working, and that's how you're productive, then go for it. If you really are okay with that kind of a lifestyle and that's how you are, then that is what you should do. But this is just so off-putting to people, to the rest of us, really.
Quinones has nothing against holidays. Vacations are great, he says. Take a weekend off. I just don't like that at all.
I think Richard and Alan said something very important. We should be smart and organized.
Yeah.
If you do this, you can plan an experiment and finish and do it 9 to 5. Or sometimes you can go more, but doesn't matter. But not every day, not Saturday and Sunday.
Yeah.
If you get a result and you want to finish it and pursue it, yeah, sure, do it. Follow your passion, but you don't have to do it all the time. The only thing you get if you put your nose to the grindstone is a ground down nose. Sorry, Alan.
Wow, that's a quotable quote. That's good.
Yeah, and honestly, in graduate school, I was there frequently. I would come in on a weekend, but I wouldn't spend the whole day.
Yeah, I know.
To split some cells, or that's a little different. But.
So take heart, Marcin.
Marcin is a postdoc at NCI, National Cancer Institute.
Yeah, just do what works for you.
If you have passion, you'll be fine.
Be organized, be efficient. It'll work fine.
And remember, the phrase overnight incubation appears throughout the scientific literature.
Right.
Yeah, that's right.
There's a reason for that.
Right.
He finishes by saying, thanks a lot and keep up the good work with the delightful and engaging podcast. I just suggested to my summer mentee to listen to you, and I believe he'll get virally hooked as well.
Cool.
Shall we continue, gentlemen, or shall we move to Pics? What do you think? We're at 1 hour and 20 minutes.
Yeah, probably ought to move to Pics. I mean, I could go on forever.
Yeah, we could do emails for a very, very long. We should probably do an all-email episode again soon.
Yeah, I have a lot. Well, why don't we read Clarissa's email since Nissin is here?
Since Nissin is here, yes.
And Rich, you should read it.
I should read it? Okay.
Because it has to do with your virus. Right.
Clarissa writes, Hi, Vince and Rich. Great discussion. What just happened? Did it just disappear?
I'm sorry.
What?
I just moved it up so that I remember that we read it. It should be up above.
Here we go.
Sorry.
Okay.
Start again.
All right. Hi, Vince and Rich. Great discussion. She's talking about the Kochia episode that we did. That was TWiV. What? I don't know.
I'll look it up while you're reading it.
Just to explain why we grow Kochia at 34 degrees C, that came up during the discussion. It grows better when we infect cells at 50% confluence. Actually, if we started plaque assay with 80% confluence at 37, as we usually do for vaccinia, we get no plaques at all after 11 days of infection. Monolayers must be infected at 50% confluence or even less than that. And we set the growing temperature to 33 to 34 degrees in order to keep the cells growing slowly. So I believe the virus grows better under these conditions, not because of the temperature, but because it doesn't like old confluent cells. The adjustments in growing conditions in order to plaque purify Kochia took us over 6 months of work. And yes, I agree with you that this paper has a lot of classical virology. I'm glad we stuck with this despite the opinion of most referees to remove these data and just leave the genomic stuff and phylogenetic analysis. And I'm glad there are still several virologists who appreciate this kind of assay and pass this to their students.
Shame on those referees.
Yeah.
TWiV 181.
That was the episode, 181.
Yeah. You know, unfortunately, in 10 years, there won't be any of those virologists left because the current crop is not interested in this classical virology. We have a new virologist here who hates plaque assays. He says they're too much work. He likes to develop fluorescent assays instead. You know, whereas I think a plaque assay tells you so much, but it takes too much work. So.
Clarissa was with us on this jaunt through Spain following the plaque virus conference. Yeah, it was great fun.
Good. That's why we had to read her email.
Yeah.
All right, let's do a couple of picks of the week. Yeah, you know what a pick of the week is?
Yes, I know.
I'll give you a few minutes if you want to think of one.
Okay.
All right. Well, you don't have to. Let's do Alan's first. Yeah, you had this left over from the dropout episode.
This was going to be my pick last week if I hadn't vanished on you. The previous episode was all about H5N1, and I wanted to pick this then, and now I can finally pick it. The CDC has just recently released this H5N1 genetic changes inventory. So this is it's essentially a big figure, or a big table that I presume they're going to keep updating. You can download it as a PDF, and it's a list of all of the genetic changes that have been observed to date in H5N1 viral isolates. So, it's one table. It's easy to say, why are we picking a table? But the amount of work that went into this thing must have been enormous because it's got all of the individual changes, what it does to the phenotype, where it was isolated in a reference, and you can track all of that. And as I say, I presume that they're going to keep updating this as more isolates of the virus appear. And
they have the ferret mutations here.
Yep.
Yeah, they're in already. It's great. It's interesting. There are not that many.
Yeah.
That's cool. That's nice. Very good. I didn't know that existed.
Excellent.
Rich.
So if, I don't know how many are, this is probably pretty common knowledge, that if you take a Mento. It's a candy that's got a sort of a jelly bean center and a coating, and you drop it into a soda, you get an astonishing reaction.
It fizzes like crazy.
Right.
It has to be Diet Coke?
No. I think
Coke.
any carbonated beverage will do.
Okay.
As a matter of fact, there's a YouTube video where some guys experimented with different carbonated beverages. Okay.
Yeah, I think the significance of Diet Coke is it's less sticky when it sprays on everything.
So it creates this huge geyser. And if you look on, as a matter of fact, I got a link to, okay. So this becomes a science pick because the question is, why does this happen? And there's a nice Mythbusters YouTube video that demonstrates that the reason it happens is because the candy coating on the Mentos has sort of micro pits on it in high concentration, and each of the pits is a nucleation point for making gaseous CO2 out of the dissolved CO2 in the drink. So it's because it's not a chemical reaction or anything like that. It's a physical reaction between the surface of the candy and the dissolved CO2 in the drink. And so there's a nice Mythbusters thing where they actually do the experiment with a coated Mento and a regular Mento. And then I give another link here to what is... I wasn't aware that YouTube did this. YouTube, actually, you can compile a playlist on YouTube, and this is a playlist of a gazillion different Mentos and Diet Coke videos where people go crazy with this, doing all sorts of basically...
I believe it must be Diet Coke, cannot be the sugar one, right?
You think so?
That was in the when we read this. When did we read this? Another day?
We were driving around Spain somewhere.
Yeah. Yeah.
Eating Mentos.
In the middle of Mentos.
So actually, I didn't look at it all that carefully, but I did. One of these guys on the playlist is a couple of, I think, high school kids investigating the reaction with different beverages. And I didn't look at that from beginning to end, but I think it addresses whether Diet Coke is really a critical reagent or not. So I don't have the answer to that, but I do know the experiment has been done.
Yeah.
Cool.
So there you go.
Thanks. So you were doing this in Spain, right?
Well, we didn't actually do the experiment. We were
just looking at the videos.
We were eating the Mentos and discussing
it.
Sounds to me like you need to go do the experiment.
Yeah.
I think so.
Try it. My pick is a FAQ called The Snake Fight Portion of Your Thesis Defense by
This is great.
Luke Burns. That's as a FAQ should be, a series of questions and answers. And this has to do with that part of your PhD defense when you fight the snake, as we all know.
Yes.
Do I have to kill the snake? Does everyone fight the snake? Does my thesis advisor pick the snake? No, your advisor just tells the guy who picks the snakes how good your thesis was.
Yes, the better your thesis is, the smaller the snake will be.
So it is a silly little thing, but those of us who have done defenses will find it amusing.
Yes.
And
I found it profoundly weird.
You think?
Yeah. But good.
But you can still appreciate it, right?
Oh, sure.
Nonetheless, okay. This was actually brought to my attention by Matt Freeman on Twitter. Indirectly, he didn't say it was a pick, so I stole it and made my pick of the week.
Good. What I like is he's got all sorts of suggested reads about other snake issues.
Yes.
Here.
Yeah, that's right at the bottom. Suggested reads. This is great. The last question is, the whole snake thing is just a metaphor, right? The answer, I assure you, the snakes are very real. That's a good episode title, The Snakes Are Real.
The snakes are real, yes.
Nissin.
Yes, I have, as a matter of fact, 2 books as a pick of the week. One is by Tony Williams. I don't know if you have done this here. It's called The Pox and the Covenant. Matter, Franklin, and the Epidemic That Changed America's Destiny. So this is the story. So Cotton Mather was the guy who was one of the prosecutor of the witch of Salem. Mm-hmm. But when on the early 70s, one of a ship arrived in Boston, and one of the sailors have smallpox, and start epidemic over there. And he was the one that start doing the viralization, so getting the pox and start doing the not vaccination but viralization on the people against all of the high authorities that wanted to crucify him. And interesting, one of them that was against him was Benjamin Franklin and his brother that said this was completely wrong. And later they proved that he was right. So I thought the book is really interesting because at the same time he was doing the viralization, he was laying down on the floor and praying to God to help him and save his family at least. So this is the first. So the book is from Tony Williams. I think you can find this in the Amazon. And the second book, this I read also recently, is is a novel from a Brazilian author called Jorge Amado. And the book is Teresa Batista, Tired of War or Retired of War. And in the middle of the book, Teresa Batista, she was like a prostitute in the northeast of Brazil, and she went to the middle of the country, and they have a smallpox epidemic, what they call the black smallpox. It was really, and it's interesting because all the doctors run away from the city. People have and left the population of the city by themselves. So the description is amazing. And what happened was that the prostitute of the city started helping everybody and doing some kind of vaccination and cleaning the disease of the people. It's really interesting. All the part of the book had described all this about the smallpox in Brazil. And so this is my 2 picks.
I see you have a theme. That's very good.
Yeah.
Thank
Now,
you.
the Amado book, I'm not finding any English translations of it. Not that there's anything wrong with that. But
I think there is a few translations.
Okay.
Jorge Amado. A-M-A-D-O.
A-M-A-D-O, right?
Yes, Jorge Amado. Teresa Batista. It's really... The book is... It's more about just one part about the smallpox that I thought was fascinating.
Cool.
Nice.
Yeah.
Thank
Great.
you. Thank you very much. We have a couple of listener picks. The first is from Gopal. He writes, Dear TWiVers, since TWiV has, from time to time, gone beyond virology to discuss how science ought or ought not to be done, I thought I would bring this article that appeared in The Scientist to your notice. Is an article called All's Not Fair in Science and Publishing. And it's an article by someone who apparently is or was in science, and his experience on someone stealing his ideas, basically.
Actually, Nissin, this is by Fred Southwick.
Oh, that's right. He's at Gainesville.
He's
Yeah,
in
he's
UF.
Gainesville, yes.
I know Fred very well.
Yeah, that's right. I saw that at the bottom. Professor, University of Florida.
Yeah.
Yeah. You know him well, yeah?
I know
Yep.
him well. Yeah.
You have a paper in collaboration with him, right?
That's right. Actually, I've published with Fred.
Yeah.
So he recites his own experience and then goes through some, scientific history is replete with these kinds of issues. And unfortunately, it's part of the culture. The bad culture of science is that people get insecure and they have to steal other people's ideas.
And he makes some recommendations for positive things that can be done that are along the same sort of lines as, collaboration and et cetera. Just a better culture. That's along the same lines as what we've discussed in our previous discussions about what's broken in science and how to fix it.
Reforming science. Gopal is a science correspondent for the Hindu newspaper in India who has often written to us. Thanks for that. And another one, we have another pick from Luis. Dear TWiVessers, a few months ago I read in Nature a few notes by Professor Robin Weiss about recent books on AIDS. One of them, The Origin of AIDS by Dr. Jacques Pepin, attracted my curiosity and I decided to read it. I have just finished it and I must say it has been an excellent read. The author, who worked in Africa many years, describes the likely events that probably started around the 1920s with the transmission of HIV from a chimp to a human and ended up in a worldwide pandemic. In the middle, many societal changes, from the colonial rule in Central Africa establishing new cities in the banks of the River Congo with mostly male migrants and a thriving sex market. Then a surge in the use of medical injections with non-sterile syringes to treat different diseases, trypanosomiasis, yaws, syphilis, malaria, and also political instability after the independence of different countries that weakened their medical systems and produced a dramatic loss of doctors, health professionals apart from teachers, engineers, etc. Highly qualified workers were brought from other countries, and in this context, the infection was exported to Haiti and beyond. Furthermore, uncontrolled blood trade in the 70s may have contributed to amplify the infection. The book is a wonderful combination of contemporary history and epidemiology and health science. Thank you very much again for your wonderful podcast and efforts to promote science in general and virology in particular, and long life to TWiV. Luis is at the Centro de Biología Molecular Severo Ochoa in
Severo Ochoa, yes. In Madrid,
Spain, where you guys just were.
Yep.
Thank you, Luis. And finally, we have a pic from Sven via Twitter. And this is a website called The Well-Formed Eigenfactor. It is visualizing information flow in science. A very cool site.
Cool.
And, different ways to put data in front of you. Really nice graphical ways to do it.
Mm-hmm.
Beautiful. Check it out. So thanks for that, Sven. And that will do it for TWiV 191. You can, of course, find us on iTunes, the Zune Marketplace, TWiV.tv. And if you like TWiV, one way you can help us is to go over to iTunes and leave a rating or a review there. That helps to keep us visible and let more people find us. And, of course, check out our Facebook page, facebook.com/thisweekinvirology. We love to get your questions and comments. Send them to twiv@twiv.tv. Rich Condit is now back at the University of Florida, Gainesville, after a long absence. Thanks for joining us, Rich.
It's great to be back.
Good to have you.
Where, by the way, it is currently raining.
Really?
We have a typical classic Florida afternoon thunderstorm going on here, and it's great. We need the rain.
A couple of weeks ago, when it was in the high 90s here, I checked out the Gainesville temperature. It was lower than here.
Yeah, I heard that. That was on TWiV. I heard that. Yeah, we had a big storm while we were out of the country that dropped a couple of feet of rain on us. So we need all this. It's good.
Here it is 29 degrees centigrade, which is quite warm, and it's completely overcast.
Yeah.
No sun. You can't see the sun. Just all clouds. Probably the same in Massachusetts, right, Alan?
Yeah, pretty much the same. It's 32 Celsius.
Alan
Actually,
Dove,
a little bit warmer.
Alan Dove is at alandove.com and Alan Dove on Twitter. Thanks for joining us, Alan.
Always a pleasure.
And your internet worked perfectly.
Yes.
Wonderful.
Yes, so we're going on almost probably day 6 now with a live internet connection.
And
I'm
it sounds
very
good.
Happy.
Bandwidth must be good because it sounds great.
Great.
Thanks. And Nissin Moussatche from the University of Florida, Gainesville. Thanks for dropping into the TWiV studio.
It was my pleasure. It was really fun being here.
Good. I hope you have a good visit here in New York. I guess you're going back to Florida in a couple days, right?
Sunday morning.
All right, and then you'll probably, first thing is you'll see Rich, right? Monday morning.
Monday morning yes.
For coffee, dude.
Yeah, Monday morning for coffee, yes.
Great, thanks again. I'm Vincent Racaniello, and you can find me at virology.ws. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.